Good morning, everyone, to today's edition of BPPV. So our first speaker today is going to be Margaret Johnson from Johns Hopkins, who's going to talk about the right timing for self-assembly. So Margaret, take it away.
All right, great. Thank you, Ashok. And I'm really excited to be able to present in this wonderful series that I've been participating in now for years, or viewing now for years. All right, so let's get started here. Okay, there we go. So macromolecular self-assembly is the primary focus of my lab's research, and really not just the structures, but really how they're controlled to assemble at the right time and place. And so this is an image, this is a sort of cartoon of a cell, and let's see if I can get this to a few examples. The cytoskeleton has to assemble to sense the environment, forming these filopodia. It assembles actually in different types of structures for cell motility. And our favorite example is there's proteins called clathrin, which are these little trimers I'm circling here that assemble this hexagonal lattice. It's actually soccer ball shape, and they curve the membrane into vesicles. And the whole point of endocytosis is to take receptors off the membrane, either because you need the nutrients that they're bound to, or because they need to be cleared off the membrane after, for example, neurotransmission. And there's another assembly process here. Okay. And so these processes are really ubiquitous, and for in part for my talk, but I think it's also a useful classification, is that there's different demands on self-assemblies, and that presents different challenges to how you actually assemble them. So these two classes are sort of macromolecular, very stable structures versus clathrin cages are two examples. And what I want to say is, for structures like the ribosome, these are very stable structures. And so we can think of the assembly process itself as kind of like a means to an end, right? You start off by translating these subunits, and then they have to get together somehow, because what you really need is this completed, stable, functional ribosome. And so the challenge here is you've got to form these structures, thousands and thousands of them, very robustly. You don't want to waste these subunits, and you have to avoid kinetic traps, which I'll define in the next slide. And so this is one of the challenges for these types of very stable structures. In contrast, for processes like endocytosis or even the cytoskeleton, we think of the assembly processes as really actually a really essential component of the function, because you want these structures to assemble only on the membrane, only when they've found receptors. And you do not want them to assemble in solution, because then they'd be empty. They would be wasting again, and they wouldn't be performing any physiologic function. So I'll return to that in more detail in the second part of the talk. Okay, so, and then we know, of course, that living systems are extraordinarily good at putting together these very stable structures. The ribosome here with 76 distinct subunits and RNA, this pre-initiation complex, so it does this over and over and over again. But if you take these subunits and dump them in a test tube, the yield is really abominable. And so this is not how living systems put these things together. It's not just a bulk assembly process. And so one of the reasons this is such a problem, if you just dump them in, is because of kinetic trapping. So we have all of our monomers here, we dump them in, and this is what we want to form, this little cartoon 7-mer that I've drawn. But of course they have to bind to each other. This is a physical process. And so they have to form all of the intermediates along the way until they get to the 7-mer. And I've sort of illustrated 4 different 6-mers that you could form. And so they're each missing one subunit. And the problem is, if you run out of monomers, so you're now starved of monomers, and now what do? None of these 6-mers can combine together to form the 7-mer. So now you have to wait. You have to wait for one of them to disassemble, and this process can be extraordinarily slow because they are all forming contacts with each other. So waiting for things to disassemble so you get monomers back can lead to dramatic delays in assembly. And also we think of this as the simplest form, the most fundamental form of kinetic trapping. We didn't allow any mistakes to be made. It's really just monomer starvation. And so in living systems in general, this doesn't have to be occurring in equilibrium. You can exploit driving forces. So chaperones, enzymes can help. cofactors. So we showed for virus assembly that if you introduce binding to another cofactor, you can delay the concentration until you get very nice assembly. Or you can imagine, especially for something kind of small, that it is actually evolved to assemble. If you dump it back in the test tube, it will actually assemble from the bulk robustly. So what we wanted to address in this study that we published last year is really how effective are these biologically inspired protocols at getting from these subunits, these monomers, to a very high yield structure and avoiding kinetic traps. What are the constraints that ensure that these work? And then what are the trade-offs of different protocols that living systems can use? So one thing I want to emphasize is that we're not trying to learn a specific structure or steady state. Those are something that we know. We're trying to learn these time-dependent pathways that will get you there efficiently. Okay, and so we like to enumerate all the possible species. So if for a trimer, for example, you have 3 different monomers, we're going to assume all of the subunits are different. And so it's not such a hard problem for the trimer, but you can imagine you have to enumerate a lot more. For larger systems, of course, we use the computer for that. And then you can write down the differential equation. So in this part of the talk, I'm primarily using non-spatial mass action kinetics, but these systems are nonlinear because of the coupled unknowns here, A and B. And so we have to, even for the trimer, we have to solve it numerically on a computer using integration. For the dimer, you can write it down analytically. So here's an example. I started out everybody at the monomer stage, and they assembled into dimers, which turned into trimers. And so here's a key phenotype of the kinetic trapping. You can see at this point, you ran out of monomers, but you still had both dimers and trimers, so you got stuck. And so this plateau is an indicator of kinetic trapping. You had to wait until the dimer fell back apart again. And so how can we quantify this? And then how can we learn ways to avoid this in these pathways? And so one way we quantify it is just the time it takes to get to 95% yield, pretty straightforward. And then the other way we quantify it is what we call a trapping factor. It tracks very well with the 95% yield, but basically it picks out these inflection points here. So you take the derivative and it finds these peaks. And this is helpful. It's a measure of how long you're stuck in the trap regime. So you entered here and you exited here. And what we did here is we optimized the system, which I'll show how it works. The 95% time is much shorter, and we collapsed these 2 inflection points to just 1. So the trapping factor is now exactly 1. Okay, so we're going to start out with the very simple model assumption. All of the subunits, whether it's a 3, 4, 5, 6, 7 are all monomers. The KDs and the free or the free energies are just a log transform apart. They're the same for all of the pairwise interfaces. And initially, we're going to assume all of the rates are equal. And what we're going to relax is that these initial conditions, we don't have to start them all off at the exact same time in the bulk, and that we don't have to allow all of the rates. So this is how we're going to tune these timescales. They don't have to be equal, but we are always going to keep the total final concentrations the same and the free energies, which means we always reach the exact same equilibrium steady state. We're just messing around with the pathways and the timescales. Okay, so first, how do we get the system to become kinetically trapped? So what I'm showing here is a hopefully clear presentation that as you stabilize the system, so the KD is a measure of how strong the interaction is, we're making it more stable, the yield goes up to 100%. And of course, as you overstabilize it, you can't get better than 100%. Everybody's in a trimer. But then at the same time, now what I'm showing in the black and the green are the timescales. So as you reach 99% yield, you accelerate. So you're now assembling very quickly. There's no trapping. But as you overstabilize it, now you see this exponential slowdown in the time to reach equilibrium. And this is because you're slowing down dissociation and you're forming these plateaus. You're getting stuck. And you can see they both track each other with the same scaling. Okay, and you might say, well, those timescales didn't look so terrible, but They actually get worse and worse the larger the assembly is. So this is for trimers and this is for the 7-mers. You can see that there's a much sharper growth in these trapping time costs. And in fact, we can show that this trapping time cost has a universal scaling. It's exponential in the size of the assembly, and that's because it's dominated by the lifetime of the most stable subunit. And so if you have a 7-mer, the most stable thing is a 6-mer, and it's very slow to disassemble that and get back your monomer. So that's controlling this, the lifetime that you have to wait to get out of the traps. Okay, so our goal is we want to avoid these traps. So we're going to optimize, again, the rates so that we can hopefully avoid monomer starvation and get nice, efficient assembly. And so how are we going to do this? The optimization, this is an optimization problem, right? We want the yield, which is the total amount of this of our desired target at a particular time to be very high. And we're going to search in the space of rates down here. So of course we want to follow the gradient to the maxima, and this is going to depend on the rates because it's not an equilibrium time that we're optimizing for. Okay, but we've got heteromers here, so there's a very large parameter space that we're searching in. And it grows as you make it bigger. So how do we search in a space with many, many parameters? We're going to take advantage of algorithms from the deep learning community. And so there's this really nice algorithm called automatic differentiation. And it's a generalization of backpropagation. So backprop allows you to train a neural network with hundreds or millions or billions of parameters very fast. Automatic differentiation allows you to train any algorithm with however many parameters very efficiently. It doesn't have to be a neural network. And it just matters that the algorithm, in our case, numerical integration, each step has to be differentiable. So you can imagine here, again, what we need in order to follow the gradient is the yield with respect to the rates. But of course, that's not what we get when we numerically integrate. We get the yield as a function of time. And so the key element is that while you're computing the yield at each time step, you might be using Euler's algorithm, you're computing it with respect to the rates. It depends on the rates. And so as you compute, you can actually evaluate partial derivatives, use the chain rule, and that's how you get the yield and the derivative with respect to the rates. And so you can follow it and iterate until you get this really nice solution. Okay, and so we have these protocols that we're designing, and we selected from 3 different protocols. One is you give me the subunits and you say, please design the binding rates between my subunits so that we get very fast and efficient assembly. And so I'm going to be the sort of, evolution process here. Alternatively, you can say evolution designed this trimer, and so don't touch the binding rates. So find some other way to get it to avoid traps. So we can design an active protocol where enzymes recycle these trapped intermediates back to monomers, or we can design a protocol where we change the initial conditions. We add in or activate subunits in time so we can control the concentrations. And so does this work? So this is to start out with, this is the bad way. This is all the rates are the same. We're kinetically trapped. You can see that these time scales are really, extraordinarily slow, longer than the age of the universe. And so what happens when we apply our protocols? You can see that they dramatically improve the efficiency. So orders and orders and orders of magnitude. And when I designed the rates, when I evolved them, these are the internal pathways. I was able to find very efficient solutions that were basically as good as this optimum you get at equilibrium assembly, where the reverse and the forward binding are perfectly matched so that you never actually have to wait for disassembly. And versus these external ones are not quite as good, but they have this advantage that they can work for any system, even if the rates are sort of poorly selected for by evolution, because maybe, avoiding traps is not your primary goal in designing the binding rates. And so just a few comments about, what are the kinds of things that we learned from this. The idea is that diverse subunits, one of our conclusions, it really helps you to find more pathways, and it simplifies the design space. So I have my 3 subunits, they're all different. And one of the solutions is And in fact, in all these pathways, you have to create a hierarchy of timescales. So you make one subunit bind fast to all of its partners. And that way, even if you nucleate everywhere, you end up with the exact number of nucleated structures that you need at the end. And so everybody else binds slowly. And so then they can add on to these nuclei that you've made, and you can get very robust and efficient assembly. You have to stratify fast versus slow. And so that's what this graph is showing. And the bigger you want to assemble something, the more you have to stratify the rates. So if you look at the gold line, you have to make it 300 times faster versus only 60 times faster. And that's because you're building something larger. So you have to wait longer to get to 7 than you do to get to 3. And the other thing that is really cool that we learned from this approach is that we only designed these systems to be very efficient, but our protocols we found were actually much more robust as well to any perturbations in the KD or the free energy. So that could happen if you mutate the subunit interfaces or the concentration. So if you change the expression. And so the Brown curve is what you get if you just sort of perfectly try to match the dissociation and association at equilibrium. So of course, if you overstabilize it, it gets kinetically trapped. That's what I sort of showed in the first plot. But our kinetic protocols never get slower. And they have a very similar response to concentration. So they're still very efficient, even if you move it far away from where it was directly optimized. And so why is that? And so we could show that it's because we've designed protocols that are completely insensitive to dissociation. So you never actually have to wait for any of your intermediates to fall back apart. And that's showing that, if you're at this optimum, the rates can be, equal to each other. As you overstabilize, you have to create this hierarchy of rates, but you don't have to keep creating a hierarchy at plateaus. And that's because, so even for irreversible binding, you can robustly assemble these structures. And so I have this sort of cartoon where the flux goes from the monomers to the dimers, and then the monomers add to both the dimers and to make trimers and form more dimers until eventually you get all of your trimers. And the key thing is I never made a backward arrow. So I never relied on any of the dimers having to fall back apart again. And that's what makes them so robust, because you don't have to tune the timescales. You just push it through a narrow number of pathways. So you don't have to make weak interactions as well. So to conclude this part, the key things that we found, these hierarchies of timescales can not only give you efficient assembly, it can be very robust. And that means that actually you need to slow down some of the binding interactions to actually get very fast assembly, which is somewhat counterintuitive. If you have diversity in your subunits, that can help in the design. You don't have to build in allostery or cooperativity in order to get these hierarchy of timescales. You just have to say A and B bind quickly and B and C bind slowly. And then even if the binding kinetics is sort of far from this perfect stratification we designed, these external mechanisms can really help you get to very high yield. So we could design in vitro protocols that would give you high yield with this approach. Okay, and so for the last 7 minutes, I want to tell you where kinetic trapping is not always a problem. If you have weak interactions, for example, you don't have to worry about kinetic trapping. And in these kinds of pathways, however, there's still challenges. And this is where we see self-assembly as really part of the decision making to help prevent assembly until you're at the right time and place. And so instead of the sort of cartoon that I advertised this talk with, this is actually a real EM image, I think from the 80s, of clathrin, this trimer protein. And you can push it to assemble in solution. And so this is a cryo-EM structure of clathrin. It's hard, though. You have to lower the pH or give it a lot of linkers to help. Alternatively, this is not what you want, right? We don't want empty. The one on the top is now an EM micrograph on this actual plasma membrane. So this is what it really looks like in a cell. And you can see it forms vesicles and flat regions. And I made these little stars because these are the receptors. This is physiologically what you want it to take up. You can't see them in the EM, but we all sort of hypothesize that this must be important for controlling when and where this happens. But in vitro, you don't need receptors. You can get very similar structures in vitro on membranes, and it doesn't need receptors. And so the regime, we think, physiologically is simply different for nucleating than it is in these in vitro systems. So can we show that clathrin assembly can wait and not just for localization to the membrane, but until you detect the membrane. So it can really be a discriminator for where and when you assemble. And so, like we mentioned, you can use active processes, but more simply, you can change the concentration, right? If you concentrate your subunits by putting them in small compartments, they will assemble. That's Le Chatelier's principle. If you dilute them, they will disassemble. But we think of the membrane as the compartment. So it creates a 2D, it's a liquid compartment that can drive diffusion between the species, support diffusion, and therefore potentially drive assembly. And this is dimensional reduction dates back to the 60s, but there they focused on target identification or search. And here we're focused on self-assembly. And so this sort of key factor that we incorporate in all of our models, especially when we eliminate space, is this dimensionality factor. So of course the volume to area is going to be important. How concentrated are you? And so, but that has units of length. So what is H? This is what makes it dimensionless. It's because when you're binding in 3D versus 2D, you have a different affinity or you have a different rate, and it also has units of length. And so there's been some previous work that characterize exactly how does the free energy or the rate transform from 3D to 2D. And we wanted to, and so what you absolutely have to capture is entropy. This is a 2D assembly of a rigid body, right? These guys are going to find each other. You can, once you're in 3D, you have roll that you can do, you can spin, you can tumble, and this is automatically going to change your free energy. You can't ignore this, but can you also have enthalpic contributions? And so we interrogated that using MD simulations. We did 3D, lipid membrane bound 2D assembly, and then we did, this is just for a pair, and then we did a 2D pseudomembrane. And what we found was indeed, it doesn't necessarily, it's not just purely entropic all the time. You can get enthalpic changes. And so what I'm showing here is the free energy as a function of the separation. And you can see in 3D, it's not very good at selecting for the dimer, but it gets more and more selective for it when you start putting it in 2 dimensions. And this is essentially an allosteric effect. You change the configuration on the surface, and then it likes to bind more strongly. And so one of the takeaways is that this length scale can actually range quite widely. We learned from comparison to some in vitro experiment, it's on the order of 30 nanometers for clathrin, and which means the dimensionality factor is greater than 1. The membrane can help you assemble. So we're going to combine simulations and equilibrium theory. This is very recent work, the theory from my postdoc Sam, to predict regimes where a receptor can tune this assembly. And so the software that we use, we've developed in our lab, it's called NERDS. It's coarse grain reaction diffusion. And so this is my trimer. This is clathrin, right? It just has these simple domains. They bind to each other and they enforce a structure. They also bind to lipids, and now they're in 2 dimensions, and the diffusion slows, although we're not going to focus on timescales here. And then it also binds a receptor. So this is the simple model that we're going to interrogate using simulations and theory. And so these are some simulations we ran a few years ago. They had one extra component, but what I want you to see here is that in the red region, there's not enough links to the membrane, so you can't take account of dimensional reduction and some built-in cooperativity to nucleate. This is the critical concentration where you can nucleate, but they will spontaneously disassemble. And down here, you have enough adapter or links to the membrane to nucleate these structures. And so the key realization here is that depending on how strongly you pull, you can drive nucleation. And so if we focus on this region here, right, you're not nucleating any of these lattices because you can't bind strongly enough to the membrane. And so that's these red curves down here. But what happens now if we start giving it receptors? Can these be tuned to actually drive nucleation? And from simulation, we can see that, yes, we added in receptors and we started nucleating coats. So this is time, and this is the size of the fraction in the largest coat. And if we look at the equilibrium behavior, so this is the size of the coat as a function of, you can see there's a transition as we add in receptors. There's no clathrin lattices here. And then eventually there's a fairly sharp transition, and then you get these nice coats. So we can show that the receptors indeed can trigger nucleation. And then I'm going to sort of zip through this equilibrium theory, but we can actually think first, because we want to have a nice formula that allows us to really explore parameter space. If you think about a lattice, we have a certain number of sites, and we put these triangles on and we can control the density of these triangles on the surface. You can define the entropy and the energy of these interactions. So we have some energy scale that we set and a valency. Every triangle has 3 bonds it can make, just like a trimer. We can use these dimensionless factors, the fraction of sites and the fraction of dilute monomers, and measure a free energy. And the key point here is that depending on how densely you've populated these things on the membrane, you will actually find the free energy minima has a cluster in it, has a Clathrin nucleation, versus here it's too dilute. And then we add in binding to the membrane using this equilibrium expression. We add in binding to the receptor. Now we have a dimensionality factor because it's 2D. And then finally, now we have the full system, right? Things can move back and forth, 3D to 2D, with and without the receptors, and we can actually predict this receptor critical concentration. And finally, we can see that the theory, which is in the lines, actually agrees really well with the simulations. And what we can see is that the key observation is that if you bind weakly to the membrane, the receptors, you have to add in to trigger nucleation, whereas if you bind really strongly, you don't need receptors. So we think of this as kind of the in vitro system, whereas in vivo, you can tune the threshold of receptor concentrations that you want to actually take up. And so to conclude, I'm going to skip that. The assembly can be controlled by driving up concentrations and using 2D. We think that we know the Clathrin parameters, and we think they really are tuned to discriminate the receptors, but the membrane cannot be too adhesive. And finally, we're happy to help anyone try to use our simulation software to study their own system. And of course, thanks very much to my funding. I'm very grateful for it while it continues. And thanks to Adeep and Spencer, who did a great job on this kinetic trapping, and to Sam and Sakao, who have been working on this Clathrin model. And thanks for your attention.
Great. Thank you so much, Margaret. That was fascinating. I'm clapping on behalf of the audience. Folks, if you have questions, please put them in the chat or raise your hand or something, and we can ask you to unmute. Let me just, we do have time for a few questions at least. Let me just begin by asking one. You talked about these external or active protocols in the first part of your talk. Could you give an example of what kind of active protocols you meant? I think I sort of missed that.
Well, there are enzymes or ATPases that can catalyze disassembly of these intermediate structures, and so that would be they would use ATP to actually bind and then destabilize the assembly. In fact, the Clathrin assembly at its very end has to be actively disassembled. Although there's evidence that these enzymes can also help during the assembly process. The ribosome, for example, there's also quite a few chaperone proteins that can bind and either allosterically or actively modify the stability of the interactions.
Right. So you design similar kind of controls in your simulations or
Yes.
in the. Okay, yeah. So Gregor has a question. Gregor, do you want to unmute and ask?
Yeah, hi. Good morning, everyone. Thank you, Margaret, for the nice talk. Actually, I had 2 questions. One is, do you have experimental data to actually test some of these simulation, or are there experiments that you could design to maybe test not the whole complex system, but a subset of that? And the second question I had is, and please correct me if I misunderstood. So you used ODE equations for basically using the mean, and I was wondering if in the real biological system you would have enough molecule to justify that assumption.
So are you talking about the kinetic trapping part?
Yes, the first part.
Okay, yeah. So those are both good questions. So for the experimental part, there's work where people have studied self-assembly of structures or they've tried to get these structures to assemble in vitro. We don't have, so there's time-dependent data on virus assembly, and we've used that to study how you can control virus assembly by basically activating the subunits because they're inhibited. And so there is experimental data we've used to actually pull out hard numbers on virus assembly. For these sort of multi-component assemblies, I mean, basically people who try to, structural biologists have, had to figure out on their own how to get these things to assemble from the subunits. And, the two examples I gave, because you can't just put them together. But as far as time-dependent data, I'm not sure. And so we do have a collaborator at Hopkins, Stephen Fried, who we want to work with, because he's looked at refolding, but also he has data on reassembly of things that you pulled out of a cell. And so we're going to work with him to get more time-dependent data on these, a more diverse array of assemblies. And then,
Yeah.
and
So
then,
about this. So I mean, people have you cited Lipovsky in like some of your slides. I mean, there's people that have done these experiments for membrane proteins.
I mean, for
And I was wondering if you could use a system like that where you have 2 or 3 proteins labeled with fluorescent dyes and use single molecule tracking to see like how they basically co-localize. I mean, with a new, I know it's really hard based on the timescales that you have in there, like down to microseconds, but I was wondering if people in the field are looking into that.
yeah, they're actually, and I have another colleague who's William Wong, who's been measuring rates of binding in 2 dimensions. And as they're actually, I mean, we also have, there's published kinetic data on clathrin assembly on membranes, and so we also use that to parameterize our model. So it's more like a bulk experiment, but with the model we can optimize the pairwise parameters. But you're right, single molecule is great for getting really specific data about the interactions, and so